Hi everyone, and welcome to the tutorial on enzymes and biological reactions. Today we are going to look at the role enzymes play as an organic catalyst. Metabolism is the overall chemical activity of cells in an individual. Right? This includes the synthesis of organic molecules, energy transformation, recycling processes, and the breaking down of unwanted substances. This process occurs in the body to keep the organism alive. Okay, and it involves, in biological instances, a number of enzymatic pathways. Okay, enzymes allow these reactions to proceed at a rate that can allow biological life to be created and formed on planet Earth. Metabolic reactants. So we start off with reactants, right? And these reactants become products. And through this chemical equation, this chemical process, A and B become C and D. Now this reaction in biological organisms requires an enzyme in order to proceed, okay? Otherwise it would occur at a really slow rate, all right? Now, in order for A and B to react to become C and D, okay, they need sufficient energy before the collisions will give rise to the various products that are C and D. Now, in order for this to occur at room temperature and pressure, we require enzymes to lower something that's called the activation energy. Now the activation energy is the energy required for a biological reaction to proceed, or for a reaction to proceed. Okay? Enzymes, what they do is they align the molecules, right, of the compounds A and B in the correct conformational state to allow them to turn into C and D. Okay? This lowers the activation energy, allowing the reaction to proceed with much less energy. Okay, so energy in reactions. Reactions that require an energy input are termed endergonic, and reactions that release energy are termed exergonic. What this means is that the products in an endergonic reaction have a higher energy state than the reactants, and in an exergonic reaction the products have a lower energy state than the reactants. Okay? This is also termed in two other ways. Okay? Anabolic reactions are those reactions which build molecules up. Okay, and these are always termed endergonic, where catabolic reactions are those that break molecules down, complex molecules down, okay, and they are termed exergonic. Okay, so what is ATP? ATP is a coenzyme that consists of adenine bonded to a ribose sugar bonded to 3 phosphate molecules. ATP is regarded as the energy currency of a cell, okay, and in order to access this energy, the cell or the organism cleaves the phosphate molecules or breaks the bonds between phosphate molecules, which releases energy for the organism to use in daily life. Now, the removal of this bond or the breaking of this bond is catalyzed by the enzyme ATPase, and it is termed a hydrolysis reaction. And as you will recall from the tutorial on biomacromolecules, where we talked about condensation reactions releasing water, hydrolysis reactions result in water being needed to fuse the bond to break it. Okay, so water is required in this reaction to break the bond. Once the ATP bond has been broken, all right, the phosphate bond has been broken, this energy is released and the molecule becomes ADP, or adenosine diphosphate. Okay, this low energy molecule can be recharged by adding a phosphate molecule to it, and this is an endergonic reaction. This process requires energy to reform ATP. So in metabolic pathways, there are a series of steps, a series of controlled steps that result in a systematic dismantling of a molecule to be used by the body. This controlled chemical pathway would only occur if enzymes are used. Okay, so what happens is, right, is one molecule is broken down by an enzyme, right? The product from that enzymatic reaction proceeds to become the reactant for the next step. Okay, this ensures that products are continually removed, right, which prevents the reaction from being stalled at any one step. So each step in a biochemical pathway is regulated by a specific enzyme, specific to the substrate. So what are enzymes? Enzymes are organic catalysts made of protein, okay, and what their role is to reduce the activation energy of a reaction, okay, and thus increase the reaction rate. Without enzymes, metabolism would be incredibly slow and it would not be able to maintain life. Okay, many enzymes are intracellular. They are used within the cells that produce them, and these include the enzymes involved in cellular respiration and photosynthesis, which we'll talk about in future tutorials. Okay, there is also extracellular enzymes that act outside the body in the digestive system. So, as you can see by this graph, all right, the reaction proceeding without the enzyme has a far higher activation energy than the enzyme preceding with enzyme. What this means is that in order to get from reactants to products, I need a large amount of energy without an enzyme and a very low amount of energy with an enzyme. And as you can see, enzymes are beneficial for biological organisms because we are, of course, at very low temperatures. Okay, so 37 degrees in humans, this is not high enough for most biological reactions to proceed naturally. So here's an example, catalase. All right, so what we're going to look at is catalase and its power as an enzyme. So it's found in several tissues of the body, including the liver, and its job is to speed up the decomposition of hydrogen peroxide, which is toxic for the body. Okay, adding the catalase makes the reaction 100 million times faster than what it would be without the enzyme. All right, and this is important because H2O2 is a toxic to the body. So the characteristics of an enzyme: you only need a small amount of enzyme to achieve a large reaction. Okay. They are not used up in the reaction, which means they can be used multiple times, all right, and then they will just keep used and used and used, all right. But this does not speed up the reaction, and it does not change the direction the reaction proceeds in, okay. So enzymes only lower the activation energy and allow the reaction to proceed in the direction which makes sense, okay. So if you have a large amount of product, all right, the reaction will proceed in the reverse direction. If you have a large amount of reactant, the reaction will proceed in the forward direction. Okay, enzymes are also very specific to their substrate, and this is an example of molecular specificity. They target only the specific thing that they are made to target. Okay, they are very substrate specific. So, as I've talked about before, they're substrates. Okay, enzymes have a three-dimensional structure. Now, this three-dimensional structure consists of an active site, and this is the part of the enzyme that interacts with the substrate. The substrate being the molecule the enzyme is trying to turn into its products. Okay. So how do enzymes bind with their substrates? They bind the active site of the enzyme and the substrate through two theories. Okay. The current theories are the lock and key model and the induced fit model. So with the lock and key model, it's pretty much self-explanatory. The enzyme has an active site that exactly fits the site of the substrate, or the shape of the substrate. The enzyme comes in, the substrate fits right in, it changes it into the products, and the reaction proceeds in the forward direction. All right. Where with the induced fit model, the enzyme is a ball, like an inert ball. The substrate comes along, sits on the enzyme, pushes into it, makes a mold into it, and this changes the shape of the enzyme, okay, which then allows the reaction to proceed. There are a number of cofactors and coenzymes involved in enzymatic pathways. Okay, many enzymes require the presence of a cofactor in order for the reaction to proceed accordingly. Okay, these non-protein parts, right, are called cofactors or coenzymes, so they're non-proteins, they're not made of protein. Okay, cofactors are inorganic molecules, and they include metal ions such as iron, calcium, copper and zinc. Okay, the best example of this is hemoglobin, a globular protein, requires iron in order to function. Okay, and it's an enzyme. Cofactors are organic molecules. They are often derived from vitamins. Okay, they assist in binding to enzymes or by functioning as carriers of electrons and protons. So, factors that affect or influence enzyme activity include pH, temperature, inhibitors, enzyme concentration, substrate concentration, cofactors, and coenzymes. pH affects enzyme activity, right? Either end of the scale. All right, so the optimum pH for an enzyme is at which the enzyme shows maximal activity. Now, both basic and acidic conditions can impact an enzyme that occurs optimally at a pH of 7, which is neutral. All right, changing pH affects enzyme function purely because it breaks the bonds between it breaks hydrogen bonds. Right? This breaking of hydrogen bonds changes the 3D conformational shape of the enzyme, thus rendering it useless because the active site is now out of shape, which means it cannot bind its specific substrate. Okay, so as you can see here, we have various proteins all working optimally at different pHs based on where they're used. So urea or urease is found in urine, okay, and it works best at a more basic pH, where pepsin found in the stomach works best at a lower pH. All right, this is because these enzymes are suited to different biological conditions. Temperature. Okay, so temperature affects enzyme activity. Now, getting straight to the point. Cold, okay, only lowers the activity of an enzyme. It does not inhibit its function. okay, where heat causes something called denaturing, okay? So heat, right, if it gets too hot for the enzyme, it will actually denature it, which is breaking the bonds, rendering the enzyme permanently useless. However, up until a certain point, heat makes the enzyme more effective, okay? So applying heat to a reaction makes the molecules move around more, which makes the enzyme more effective. However, if this heat gets too great, so say in humans, past 40 degrees, the enzymes start to denature, and this process means the active site becomes rendered useless because the hydrogen bonds are being broken, all right? But at low temperatures, the enzyme just lose kinetic energy, which means they're just not as effective, okay? So here's three different enzymes found in dogs, perch, and trout, okay? So the body temperature of trout is 10 degrees roughly, optimally, yeah? In perch, it's 20-something degrees. In dogs, it's about 35, okay? So you can see how these enzymes have evolved over time to work better in different body temperatures. So, temperatures, right? As I talked about before, if the protein gets too hot, it will become permanently damaged. This permanent damage is termed denaturing. In contrast, all right, enzymes are not denatured when it is too cold, okay? They just become inactive at low temperatures due to low kinetic energy. Inhibitors. Now, an inhibitor is any chemical that changes the shape of the active site of an enzyme, so it has low affinity to the substrate, okay? They can be reversible or irreversible. Reversible inhibitors are used to control enzyme activity as they only temporarily deactivate the enzyme. Heavy metals such as lead, mercury, and arsenic are toxic because they are irreversible inhibitors of enzymes. Okay? Inhibition can be competitive and non-competitive, or also allosteric. Different types of inhibitors. So competitive inhibitors inhibit molecules that compete with the substrate for the active site. So they're actually in direct competition with the enzyme, okay, for the substrate. They slow down enzyme activity by blocking the substrate binding to the active site. Non-competitive inhibitors allow the substrate to bind to the active site, okay? But they slow down enzyme activity by binding to the enzyme somewhere else. So they bind to the enzyme in a different spot to the active site, right? And this prevents the enzyme from actually functioning properly and catalyzing the reaction of the reactant. Allosteric inhibitors do not compete for the active site. All right? They bind elsewhere to the enzyme in a manner that causes a change in the conformational shape of the active site, such that the substrate will no longer fit into that active site. So the next factor affecting enzymes is enzyme concentration. So if we have unlimited reactants, okay, and we increase enzyme concentration, the rate of reaction will subsequently increase in a linear fashion. Okay, increased enzyme concentration will increase the rate of reaction if you have enough reactant. Substrate concentration. So, all right, if we have a set amount of enzyme and we increase substrate concentration, the rate of reaction will plateau off. It will plateau at a point where every enzyme is being used in that reaction. Okay, so at a set amount of enzyme, once the substrate reaches a certain level, the rate of reaction will not get any higher. This is because every enzyme is constantly being used and the active site is constantly being filled. Okay, so the rate of reaction plateaus and proceeds accordingly based on increased substrate concentration. So, two very important chemical reactions in life on Earth include photosynthesis and cellular respiration. Photosynthesis is an endogonic reaction and cellular respiration is an exogonic reaction. Both require multiple enzymatic pathways, okay? And in subsequent tutorials we are going to look at them. I hope you've enjoyed this tutorial on enzymes. Thanks.